watching this video, please share with all your friends. And if you have time, fill in the brief survey on the website to help us with our funding applications. We'll have a short presentation by Tanis, followed by a question and answer. And then we will also record and post this session on YouTube for you to view later. And with that, I will mute and start.
Welcome, everyone. Thanks for joining us, and thanks, Mr. Wist, for having me. This presentation is on Medical Laboratory Technologist, also known as MLT. Next slide. I'm Tanis Tomaszewski, and I'm a Medical Laboratory Technologist in microbiology at the Dr. F.H. Wigmore Regional Hospital. Next slide. As an MLT, there's 5 general areas of study. There's clinical chemistry, hematology, histology, transfusion science, and my favorite, clinical microbiology. So when I told my family that I was going to study MLT, most of them giggled and said, "Oh, you're going to be a vampire, you're going to be taking blood." And while taking blood is a very small part of our job, in some places you actually won't even take blood as an MLT. So if that interests you, studying for a lab assistant or a phlebotomist would be an area that you would go into. You can see on the slide, there's a picture of a few tubes of blood. You can see they have different colored caps. And so for different tests that are ordered, they require different preservatives and different ways to keep them until they're put onto analyzers. So that's why there'll be different colors of tubes. And we'll talk a little bit more about that later. Next slide. So chemistry is the first one we're going to talk about. This analyzer here, it's kind of deceiving in the picture. It's actually quite large. It's called an Architect. And it's probably the size of a small car, maybe not quite as wide. It's like 10 feet long. It's quite tall. If I was having to do any repairs or add things, I usually need a stool to get inside of there because it's quite tall. And in chemistry, we can run many different tests. Usually tests like electrolytes, glucose, that would help to monitor a patient if they had diabetes. So in our lab, almost all of our analyzers, we have 2 of them. So we have 2 of these Architects. So it takes up quite a bit of space. All the analyzers are hooked up to computers. They're interfaced so that the computers talk to the analyzers, letting them know what tests need to be run. So when you get your blood drawn, we type it into the computer and a set of labels comes out with your name, your health number, your birth date, and it gives it a personal barcode. And it also has information for which analyzer, so the analyzer will know exactly which test the doctor has ordered just for you. We will get the sample prepared, and so for instance, the tube on the right-hand side, it is called a serum separator tube. And so when the blood goes in there, we allow that to clot for 20 to 30 minutes. And then we put it in a machine called a centrifuge. And it is spun down, and you can see at the very bottom, it looks black, but those are all the red blood cells. In the middle, there's a gel separating. And then at the top, that is called serum. And so in chemistry, most of our tests are run on serum. So these really large instruments can run about up to 300 different patients, and each patient may have 15, 20 different tests. So these machines are quite amazing on what they can do. Next slide. So next department is hematology. On the left-hand side, there's a machine called Coulter DXH, and that will run tests called CBCs, which are complete blood count. So you may have learned in some of your classes at school about blood cells. There's the red blood cells, white blood cells, and platelets. The red blood cells carry oxygen to your body. The white blood cells help fight infection, and platelets help in the clotting process. And so this machine will count them to make sure that your numbers are normal. And so when the results, and same with the architect, when the results come off the machine and onto the computer, the MLT will review them. And they'll make sure that they are don't need further testing. Like they may need to be repeated. They may need, if they don't make sense, that perhaps they may even need to be recollected. So that's part of the MLT job, is to know all the factors that would interfere with making the results accurate. So on this DXH machine, if the machine flags something for us, we will take a tiny drop of blood from the tube and we'll put it on a smear. We'll let it dry, stain it, and then we will actually review it under a microscope. And sometimes we need to do manual counts to confirm what the machine actually has given us as results. The machine on the right actually is called a top, and not every lab will have this machine in their hematology department. We actually have ours in chemistry, but this is, I thought it was a good place to talk about this. It is for coagulation. So maybe your grandparents or someone you know maybe is on a blood thinner. So this would be the machine that would test the parts of your blood to see how your blood coagulates. So it's quite interesting. It can run about 25 samples at a time. So another reason why the doctor may ask for this test is if you're going for a surgery. The doctor will want to know for how your blood clots, if you're clotting normally when you have a surgery, or if it isn't, and they'll decide your treatment from there. Next slide. Histology. This is the only department that I have not actually worked in since I trained, but I do have lots of coworkers who really enjoy this department. This department works closely with specialized doctors called pathologists, and in our lab we have 2 pathologists. And in this area, you would, an example of a test that would be done is if a patient went in for a breast biopsy and they wanted, the doctor was looking for cancer, you would be preparing these samples and getting them ready for the specialized doctor, the pathologist, to do the tests and to read all of these samples. Transfusion science is also known as blood banking. And in some of your classes, you may have learned about doing blood types. So you might know your blood type is O positive or A negative. This is where we do that testing. And you can see that there's lots of smaller things. We don't have any big analyzers in our department, so it's a lot of manual testing. So you learn lots of background in here. It's a really technical area. It's great. Some people may donate blood, and those units of blood come to the transfusion science or blood bank, and we store them there until they're needed for a blood transfusion. So if there's been a car accident, or you have anemia, or you had a surgery and you require blood, this is the area where the testing would happen, and they would make sure that the blood is safe for you. They'll do compatibility testing. So they actually will take your blood and they will take a sample from the unit of blood and make sure that they're compatible. Next slide. And so this is my favorite area, is microbiology. So right now, that's the department that I am working in. So we do tests in microbiology as urine culture for a urinary tract infection, or a throat swab for strep throat. That's where your samples will come to this department. So a sample will come in, we'll go into a biosafety cabinet, and we will put the samples in there. We will put them on, you can see on the right-hand side, there's those little, they're called plates, and they have agar with different kind of nutrients to grow the different kinds of bacteria. And so in this department, we can grow bacteria, viruses, parasites, fungus, and we're looking for things to help the doctor to treat you. So once we put the sample on the agar, we also put it on a slide. We take the agar, and on the left-hand side, you'll see there's an incubator there, and we'll put the plates in the appropriate incubators for, depending on the sample type, we put them in for 1 to 3 days, opening the incubator daily and recording what we see and start working on any bacteria that we know is harmful to you. This incubator is a CO2 incubator. Some bacteria actually like different conditions, so we try to enhance that for them. So this incubator has a 5% CO2. We do have, which is carbon dioxide. We also have oxygen incubators. We have incubators that are 42 degrees, which some bacteria like a little bit warmer. After I told you about the slide that we prepared, we stain it and we look under the microscope at the slide as well. And we look for bacteria, and we also look for the white blood cells. The white blood cells, we said, fight infection. So we know if we see the white blood cells, we know that most likely there's an infection in that area, and that information will help the doctor. Next slide. So once we've grown the bacteria, we want to know what bacteria it is. We'll make a dilution, a very precise solution in a little tube of saline. And you can see on the right-hand side, there's that white thing, we call it a card. And on the bottom, there's a straw. So we'll put that straw inside that dilution of the bacteria, and we'll put it into this machine. And so you can see the little tiny dots on the cards. Inside those dots, there's chemicals, or there may be antibiotics. And so the chemicals will help identify, and the antibiotics will let us know which antibiotics will be suitable to treat this organism or this bacteria that you have growing. So this takes about 18 hours to run. So the next morning, the MLT will come in, it will review, she or he will review all of the results and make sure that they make sense. They may need to do a few more manual tests to make sure that these results are accurate. Also in microbiology, you will learn which organisms are normal in certain parts of the body and which ones are pathogens. A pathogen is what actually causes an infection. So you're going to learn and make sure you know the difference between that. And that's why I really enjoy microbiology, is because there's so much variety every day. And it is, for me, it's almost like growing a garden. You're growing the bacteria and you're learning it. You give them and make sure that they're growing properly, and then you identify and you help the doctor. Next slide. Maintenance is very important in every place in the lab. So I just showed you a few of the analyzers that we have in our lab, but we almost always have 2 of our analyzers in case one gets broken. So we need to maintain all of these machines. We need to change parts, we need to change solutions, reagents. There's daily maintenance, weekly, because we want to make sure that these machines are working the best that they can, because we don't want them to break down, because that will cause a delay in getting results out to the doctors. We work on computers all day long. So all of our machines, almost all of our machines are hooked to computers, so that you spend a lot of time with that. Microscopes. You will probably spend at least part of every day on a microscope, and some days you might spend most of your day on researching with the microscope. And quality control. It's a big part of our world. We want to make sure everything that we are sending to the doctors is accurate. So we have samples that have known results, and we run those samples sometimes once per hour, sometimes once per day, depending on the machine and the test. And the results have to come out to that known result. And if they don't, then we won't release any results to the doctor, because we want to make sure that everything is working properly. Communication is very important. You need, every day you're calling doctors with critical results, or you're calling nurses or other healthcare providers and letting them know. Other times doctors are calling you to add tests, you're working closely in a very busy environment with lots of other coworkers, and you need to keep everything flowing. So you need to have really good communication. Attention to detail. So I'm just going to give an example. So I talked earlier about the tubes of blood that come in with a patient has been identified. There's labels that come out, and each label that has that special unique barcode also has the barcode is unique for each machine. So if you take one of the tubes and you put the wrong machine label on it, when you put it on the machine, it will say, sorry, I don't run this test. But when you are running 100 tests at a time, it might take you a little while to figure out why that test or that tube of blood hasn't been run on your machine. And so this would cause a delay, and that's just because you put the label for this machine on the other one. So it's very, very important that you pay attention to every little detail, because you don't want to cause delays in the doctor getting results for the patients. Multitasking. There is lots of things going on at once. With this job, there is shift work involved, and sometimes you are responsible to work in 2 or 3 different departments, or maybe the whole lab. And you need to be able to know all the tests that are running at one time and get them all done in a timely fashion. You make about, as an MLT, around $63,000 to $69,000. You may make a little bit more because there is shift work, so you get paid a little bit more for shift work and working weekends. Benefits and pensions are with the union. I'm with SEIU, and there's also a QP union for MLTs in the province, and we make very similar wage, and our benefits and pension are the same. You start off holidays about 3 weeks a year, and with your years of service, you can get up to 6 weeks vacation. Our shifts are 8-hour shifts, and we do have shift work. So this is a big part. Almost all MLTs work shift work. There are a few positions that don't have shift work, but this is a big thing. So you may be working days, you may be working evenings or midnight shifts, and you also will work the weekend, because the lab is open 24 hours a day. Even though they don't take blood from patients, outpatients, 24 hours a day, there are still people in the hospital and emergency rooms that need that work done. I am licensed with the SSMLT and the CSMLS. So SSMLT is Saskatchewan Society of Medical Laboratory Technologists. So that's required for this province. Being licensed with the Canadian Society of Medical Laboratory Scientists is optional at this time, and I am licensed that way. You need working hours and continuing education to maintain your license, and you pay this yearly. It's just a little over $400, and the continuing education credits are quite easy to get. You just need to keep on top of it. There's always opportunities from the societies. There's workshops or planned events for you to attend to gain your credits. Sometimes when your lab gets a new analyzer, they come out to train you on that analyzer, and you can get credits that way. Or you can find continuing education credits lots of places, but you're always continually learning in this field. You're an important, vital part of the healthcare team. And as we are in COVID right now, the MLTs test for COVID. And so you can see how we are an important part of the healthcare team. How many tests are ran on COVID are reported daily. So it's an important part of the healthcare team. The work-life balance for me has been probably the biggest challenge with shift work. It's important that you maintain your health and your sleep and your relationships with friends and family, because when you work shift work, you are sleeping when people are at work, and you're at work when everyone else is at home. So it's sometimes hard to connect. So you actually have to make an effort to make sure that you stay connected for that healthy lifestyle. So I just want to thank you for listening in, and if you have any questions, I'd be happy to answer if I can.
Great, thanks, Tanis. That was wonderful. I guess kind of your training, how long did it take to be your training to become a technologist?
So right now the training is in a change. And when I was looking up the exact time, the Sask Poly website has been down. And so and it's with COVID, they're changing it too as well. So when I went to school, it was 2 years, but that was a few years ago. And I do know they've extended the program and it can be up to 3 years. It's usually about 1 year of in-school training with labs, and then you go to a practical side, to a hospital to train with some in-class sessions.
What would be the biggest challenge with your job? Like the thing that is the most challenging part?
I would say being on top of your game every single day, because what you're doing is very important and there's no room for error. You have to be on your game every day.
You talked a little bit about the COVID testing. That is something that you do in your hospital, in your lab?
Yes, we do a little bit of that COVID testing. Because of SHA and privacy issues, I don't discuss too much of it, but it is something that MLTs do, and it's very important and high priority list right now.
Awesome, thank you.
What kind of things do you have to do to maintain the machines?
So maintaining machines, we, like lots of the machines have pipettes and probes to aspirate samples. We change the pipettes, we clean. Lots of different areas of the machines require different reagents for cleaning. So like one area of a machine will require 3 different reagents in a certain order to clean. And then you move on to another area and you'll clean something else with 3 or 4 different chemicals. And so, yeah, it's important that you can follow steps and procedures carefully to make sure that all of our machines are working well.
So that is part of your description, is to make sure the machines have been maintained?
Yes, every day we do maintenance on all of our machines. Yes, so when you come in, that's a big part of your training, is learning machines. And each hospital will have different machines. So that's the other neat part, is that you will learn different analyzers at different places. And this is one other thing I wanted to mention, is Moose Jaw is a unique place to work. Lots of new graduates like to work here because when you're done school, you're maybe not sure of which department you really enjoy working. And so when you come to Moose Jaw, there's the core area. So that's 3 different departments: chemistry, hematology, and transfusions. And that would be the main area. And then some people will get trained in either microbiology or histology. If you worked at a big facility, perhaps in Regina at one of the hospitals, or in Saskatoon, you would likely be hired into just one single department. And you'd be hired, say, into chemistry, and that's all you would do was chemistry all day. When you come to Moose Jaw, you get that variety. You get to work in the 3 main areas and get that experience and kind of see what you enjoy doing. And some people like to stay here because they like that variety.
You have a favorite area that you like to practice, and why?
And microbiology is definitely my favorite. I think it's because it's like growing a garden. You see something different every single day, and it's a lot of hands-on. Whereas we just have that like one machine that we use. A lot of the other testing is manual and interpreting of the results. And so I enjoy that.
How big is your department? How many people would you work with normally in a day, or in a shift, I should say?
Yeah, so it depends on the shift. But say on the day shift, like including supervisors And we also do have phlebotomists and lab assistants that work with us. I would say about 25 people work with us maybe on a day shift. The evening shift goes down to 3 people. And then on the midnight shift, it's down to 1. So you can see on the midnight shift, there's a lot more responsibility that the hospital relies just on that one person.
Is there a huge demand for your areas, or are you seeing a lot of grads?
Yeah, there is a big shortage for MLTs right now. And so, yes, it's a great place. There's job openings all the time, and there's still lots of people that are eligible for retirement. So yeah, there's openings everywhere in the province right now. And especially during COVID, there's a bigger demand because there's lots of people in Regina, Saskatoon, that their job is just dedicated to COVID. So there's been that big increase as well.
Your presentation was awesome, Tanis. I loved seeing all the machines that you use and you know, the description of each one. And I can definitely see the difference between a technician and a technologist now too. So thank you.
Thank you so much, Tanis, for sharing with us today. I'm sure the students will get a lot of valuable information when they watch the YouTube when trying to determine their career.
Okay, thank you for having me.